there are some people here who haven't been here before. Am I right? Is there anybody who has? Is this the first time you're here? Okay. So, Jeffrey, what is this a picture of? And why would we have a picture of the Brooklyn Bridge in a course called Demystifying Medicine?
Maybe trying to bridge from science to clinical medicine.
You got it. Okay. So this is.
Not get the bends in the process.
Pardon?
Not get nitrogen narcosis in the process.
That's correct. Yeah. So believe it or not, this picture was actually taken by my grandfather, and it is the Brooklyn Bridge. And the point is that the purpose of this course is to put us, like the two gentlemen on the catwalk, and we're connecting Brooklyn and Manhattan. Now, it's important that when the bridge was proposed, the people in Brooklyn, they objected. They didn't want these city slickers coming into their rural area. And the people in New York said, we don't want those country bumpkins coming across and spoiling things in Manhattan. And of course, they built the bridge and everything flourished. Now, there's a moral to this story, because by connecting the incredible advances in basic biological and engineering sciences with major health problems, it's not so easy because everybody speaks a different language. So one of the purposes of this course is to have people communicate. And by the way, if people are using a language you don't understand, technically speaking, speak up and ask them to define what they're talking about. Because I think that the lack of this communication is probably one of the most difficult, serious problems that we confront. I like to think of science as having progressed in the past 25 years as almost logarithmically, and its so-called application to human health, or for sometimes even human physiology, you can think of as arithmetic. And so this gap gets bigger and bigger and bigger, and just throwing money at it doesn't solve the problem if people don't speak the same language. So we're about to go between Brooklyn and New York and backwards and forwards, and the topic today of HIV is one that we have had as part of this program here at NIH, I think in 11 of the 12 years that we've been doing it. And during that period of time, almost everything has changed. The epidemiology has changed, the public awareness has changed, the science has changed, but the problems still remain. They remain in the science, and they certainly remain in terms of public health and knowledge and so forth. And in the global aspects of it, we now have superimposed, the major centers in the world where HIV is rampant, particularly in Southern Africa. So the problem as a biological challenge is as challenging as it was almost since the day that the virus was first discovered. And as a public health challenge, although it's been changed to a treatable disease, with the exception perhaps of one individual on Earth, no one has ever been cured of HIV. So we also have a bit of a celebration today, because the first speaker, my friend and colleague, John Coffin, this is the 20th year that John has been the Eastern Airlines professor of microbiology, molecular biology. Now, what do I mean by that? Well, John, we were faculty members at Tufts University in Boston, where John still is. He's an American Cancer Society professor at Tufts. And 20 years ago, he was presciently recruited by Harold Varmus to head an HIV drug resistance program here at the NIH. And he has dutifully been commuting back and forth. He's outlived Eastern Airlines. Now he's on National Airlines, I think. But this has gone on for 20 years now. And enormous changes have taken place in which he and his colleagues have been major participants. John is one of the world's experts in retrovirology, and I think certainly one of his major contributions to HIV is the recognition that drug-resistant forms of the virus are present from the very beginning. At any rate, he will describe this and discuss other, and I'm sure, exciting things that have developed in the past 2 years, because last year he had a vacation and didn't teach in the course. So it's really a great pleasure, as always, to have John Coffin take part. John?
Thank you very much, Win. It is indeed a pleasure to come back yet again to give this course, give this lecture, and a great pleasure to do it for the first time with my good friend and colleague Jeff Lisson. We're both in the same building, although somewhat different, rather different employees. We're both actually neither of us is a government employee. We're both contractors, in fact, so you can take that for what it's worth, I guess. So I'm not going to talk to you today about drug resistance, although that indeed was how we got sort of started setting up this program, which we now call the HIV Dynamics and Replication Program. That's because the focus has turned away from drug resistance somewhat, but we had to keep the same acronym, so that was very important to me. So I do want to talk about what it is about HIV. One of the medical miracles of the last part of the 20th century was the discovery of antiretroviral therapy that could almost completely eliminate the progression of HIV infection to what had been the inevitable death sentence of AIDS. Until that time, within about 20 years, virtually every person who was infected with HIV would die of AIDS if they didn't die of something else before that. And when modern combination antiretroviral therapy came along, that actually brought that death rate down to close to zero as long as it was used properly. However, there's a the downside of that is that the therapy is not curative. It, in fact, if you if you monitor HIV infection, then the best way to do that is by monitoring the amount of virus that's been released into the bloodstream. And the reason that that's a good indicator is that virus that's released into the bloodstream is an indicator of the virus that's infecting cells elsewhere, probably mostly in tissues. But it turns over very, very fast. So that at any given time, the amount of virus that you see is in fact a good measure of the amount of virus that's being produced. And that's a good measure of the number of infected cells. And when you start therapy, before you start therapy, the virus remains at a pretty good steady state level, usually averaging around 20 or 30,000 copies per milliliter of RNA, which you can do using PCR. You have good quantitative RNA measures. When you start therapy, that virus drops very rapidly and goes down by several logs to a level which you can no longer detect it with a standard clinical assay, which is 25 to 50 copies of RNA per milliliter. But if you use more sensitive assays, the level, you can find that the level actually of virus remains detectable with the more sensitive assays, dropping down and then leveling off to a level of maybe 3 copies on average, with a big variance around that average in multiple phases that I won't go into the reason for. And this will remain pretty stable in a patient for many years or decades even. But any time that after this, and almost every patient that's been looked at, if you stop therapy, the virus comes right back again. It comes back pretty much to the level where you started before. So there actually is both a reservoir of some kind of virus that allows the virus to come back, and also a memory of the host-virus relationship, so that the level actually returns to very much like what it was before. If you monitor instead of the RNA, if you monitor the number of infected cells, and HIV is a retrovirus, and a retrovirus replicates by copying its RNA into DNA and causing that DNA to be integrated into the cell, and I'll tell you a lot more about integration later on, then you find a rather different kind of curves. It drops a bit, maybe 10-fold or so initially, and then levels off at a much higher level than the RNA relative to what you started with. And that means that although you have a 4 or 5 log decline, a 10,000 to 100,000 fold decline in RNA, you only have about a 10-fold decline in DNA. And that means that of this DNA, the fraction of this DNA that's devoted to making the virus at this point is much, much less than it is here. And that's actually another instance of an adage I like to use, which is in HIV cure research, and I think it's also true in HIV vaccine research, is that the better the science, the worse the news. Because that means that, first place, the virus infection cannot be cured, and it cannot be cured clearly because there's this persistence of DNA, but also, and this has been very elegantly shown by Bob Silicano's lab in Baltimore a few years ago, the amount of DNA that you see here is not a very good measure of the number of cells that are capable of giving rise to the virus, because that's only a very, very small fraction of these cells that have DNA in them is the virus that will give rise to this virus. And in fact, what Silicano lab showed was that only a very, very small fraction of this DNA is even intact. Most of the DNA that you measure is defective mutants of various kinds that have found their way into very long-lived cells. These are immune cells. These are CD4 positive T cells. Their job is to respond to an antigen and then go into a latent memory state, which then can last and survive for years and years so that the immune memory comes back. The virus has very cleverly found its way into cells which can expand very rapidly and make lots of virus, and then can go into this resting state and live for very long periods of time, rendering it effectively incurable by agents that simply block virus infection, such as standard antiretroviral therapy. So what's going on during this time? What is the nature of the reservoir? Again, the Silicano lab first showed, and we've confirmed, and this shows an extension of that kind of experiment, that here's another, this is now a real patient. With time on therapy, you can see the drop in the viral, the level of virus in blood, and you can also see the smaller decline in the viral DNA in the same way that I showed you before. And what you see is during that time, if you follow the genetics of the virus and by making these phylogenetic trees, what you find is that with time, if you look at the earliest samples, the virus that you see in those early samples is very, very diverse. There's no two sequences are alike. However, with time, what you see is two things. One is that the virus does not evolve at all. These branches, evolution will be measured by these branches getting longer, and I'll show you a case later on, which is a kind of a positive control for this kind of experiment. Instead, the virus remains in evolutionary stasis. A property of retroviruses and HIV is that as they replicate, they accumulate mutations. And you can use the accumulation of those mutations to tell you whether real replication of the virus is going on. What you do see, however, is not the accumulation of additional mutations above what was already present in the diverse population that was in this patient, but you do see the appearance of these rakes, these groups of absolutely identical sequences that indicate that something has happened to cause a clonal expansion of one particular virus genotype. And a large part of what I'm going to tell you is to try to convince you that this clonal expansion is not due, it could have been due to just a virus breaking through and then clonally rapidly outgrowing everything else in the body. But the evidence is very strong that I'll show you that it's not due to that. It's due, in fact, to the expansion of a single cell, which was infected with virus probably before therapy started, and then that cell has expanded as probably as what it would ordinarily do as part of the immune system. That's what immune system cells do. If they encounter antigen or if they encounter homeostatic signals, they will divide and can divide into rather large clones, particularly in the case of a new antigen. So, and so in fact, you could see this, again, you can see these rakes of identical sequences. This is now 4 patients showing the same kind of thing. And these appearance of these identical sequences and the failure of new genetic distance to occur, where these open circles are pre-therapy and the closed circles are after a long time on therapy. And you can see again, these plots at the bottom are just a way of measuring the amount of genetic variation that's occurred with this time. And if the virus were replicating, I don't have a control for this, but if the virus were replicating, these plots would look like this. Instead, they're absolutely flat. And that also extends to the virus that comes back. If you stop therapy, in this case after 7 years, in this case after 5 years, the virus that came back, again, had no additional accumulation of genetic distance. That's the virus as shown by in this case, orange circles, but in fact did have the same kind of clonal outgrowth. So what I was showing you before for the bulk DNA or the bulk RNA in cells is also true for the infectious virus in this patient, the virus that came back once therapy was stopped. In the examples I just showed you, we started with the patients who'd been treated in chronic infection, so the virus is already very diverse. Just to show you one example which shows that much more clearly, we also recently did a study with some babies in South Africa. You don't get infected babies in the US anymore, thanks to preventive therapy. And who had been treated at a very early age, were born infected, had been treated at a very early age, and their virus was actually much less diverse. This first example shows a patient where the sample was taken at a couple of months of age and therapy was not started until much later. And then the sequences were examined at the age of 8. And in that case, you started out with much less diverse virus, as in the colored circles here. But you could see the appearance of evolution of the virus, and that we infer occurred during this time before therapy was started. So that serves in a sense as a control, showing what happens when you do have some ongoing replication. However, if you have good suppression, what you find, and this is a patient now who was suppressed, whose first sample was taken at the time antiretroviral therapy was started very early on, and then treated for 8 years. And this shows that the virus, this is the identical. This is probably the virus that the patient was originally infected with. Most patients are infected with a single virus particle. So early on, the virus that spreads in the patient is clonal before diversity starts to accumulate. A little bit of diversity accumulated. And when you look at 8 years, after 8 years of treatment, you find this same sequence is still present and a little bit of diversity, very much like what you started with. So there's been no evolution whatsoever going on in this patient. I emphasize this point because there's a paper that appeared in the literature about a year ago, which is completely wrong on this issue. It's very badly flawed. And we're trying to get a rebuttal published as we speak. It's been a real uphill slog. Don't try to do that. It's sometimes not worth the time, but it is nice to try to clean up the literature. So there's no evidence that we can find for a role of HIV replication in maintaining the reservoir. So what does maintain the reservoir? So what we visualize is that before you start therapy, you have a mixture of short-lived cells. Cell gets infected with the virus. The virus replicates in that cell. That as part of the as part of the replication or part of the immune response or both against the virus, that cell gives off a burst of virus and then dies within about a day and a half or so on average. But during this time, a very small fraction of cells get infected, and these cells go into some kind of a latent state, and it's probably cells that were activated here and are in the process of transitioning back into a memory state. These are immune systems. These are CD4 positive T cells by and large. And these long-lived cells then have the provirus expression very strongly suppressed. And on antiretroviral therapy, these get stripped away because they no longer get infected and they die of their own of the infection. These, however, will survive. And two things can happen. One is that they'll gradually be eroded just sort of by natural processes, or maybe because they occasionally make some virus and die from that respect. But at the same time, there will be of others will actually undergo clonal proliferation, and this would give rise to the rakes of sequences that you see, and pretty much maintain the numbers of total numbers of infected cells in a sort of a pseudo homeostatic process. So the number of infected cells after a while remain about the same, and you could see that from the flat DNA curve I showed, but clonal populations of virus appear. And so to look at this in more detail, we developed or improved on an assay for looking specifically at integration sites. I'm not going to talk too much about integration, except that HIV DNA can integrate at many millions of possible sites. We estimate that there are about 10 million possible sites for integration in a cell. Only a small fraction of the genome, but a lot of sites. So the probability of it happening twice in any given sample of reasonable size independently is very small. In long-lived cells, sites of integration are determined both by initial preferences, but then they can also be modified by selection or by chance, by the lucky one that integrates into a cell that's going to respond to some antigen, is going to therefore divide like crazy. So we have, to look at this, we've looked at integration preferences in different kinds of cells. I'll show you the example of just PBMCs of white blood cells and HeLa cells, a very aneuploid cell line in culture. And this just shows the distribution of integration sites across a chromosome. I picked chromosome 16 here for reasons that will become clearer later. And you can see that there are very strong hot and cold regions. The scale here is megabases. This chromosome is about 80 megabases, 80 million bases long. And this just shows the accumulated integrations within 1 million base pair chunks of that sequence. And now if we take a small fraction of the genome, which shows a bit right here, and expand that, so now we're on a scale where we can see individual genes, we can see that there's a very clear integration pattern, as was shown actually by, among others, Dr. Levin, who's sitting in the front row here. The integration very strongly favors genes in these cells, and so you can actually really determine where and express genes. Well, I don't show you that here, although so you can pretty much determine where genes are by looking at integration patterns. So it's kind of perhaps a hard way to do it. And you can also see that these three types of cells, HeLa cells, which are the aneuploid cell line, the PBMCs, this would be actually just in the CD4 positive cells, although we didn't sort them for this experiment. You can also see the same pattern, somewhat smaller numbers of hits, but the same pattern, and the same pattern in PBMCs, which were either whether or not they were stimulated by adding PHA to them, an activating agent that stimulates the cells to divide. And in fact, we've looked at a number of different cell types. There's a very, very strong correlation of integrations. Almost all of the integration sites from one cell line, type of cell to another, and the preferences are very, very similar. You can see one case in HeLa cells of a gene that's hit, that's not hit in these others, and we presume this is one that's expressed here and not in the others. So, so in long-lived HIV-infected cells, sites of integration are determined both by initial preferences, that is the kind of hotspots I just showed you, and also by selection after integration. So what happens to this distribution if we look at patients on long-term antiretroviral therapy? So I'm going to spend the rest of the time actually talking about just this one patient, so we'll turn this into a case, essentially into a case report. Unfortunately, I could not bring the patient along because sadly he is no longer with us. He died about 3 years ago of a malignancy, probably unrelated to the HIV infection. And he reported to the clinical center very sick. He was down to about 10 CD4 cells per microliter. Normal for probably we have about 1,000 CD4 cells. So he was really low. He had frank AIDS. He had probably been infected. This was 1990 he had probably been infected, or I'm sorry, this is about 2000. He had probably been infected since about 1990, one would guess, just from the severity of the disease. He responded very well to antiretroviral therapy with a few what are scientifically called blips along the way. And then he had a couple of bouts. He lived actually in a rather remote region of the U.S., in the middle of the Virgin Islands, actually. And so I had some breaks in ability to get drugs. And as a consequence, had some rebounds of virus. And then toward the end of his life, as it turned out, the virus while still on therapy, the virus started to creep up, the level of viremia, although not nowhere near the original levels. And so we looked more carefully at this rebound virus, and you could see here, this is just an enlarged view of what happened. And it was clear, as I'll show you in a second, that the virus was becoming, in part, resistant to the antiretroviral therapy, so the therapy was switched at this point and went back again. This patient was also diagnosed with the malignancy that I mentioned right in here, and that we think is also related to this increase that I'll show you, and I'll show you the reason we think that. So at this point, we pulled the virus again. This is out of his blood. And when we sequenced it and put it on this tree, you could see two different populations of sequence. One, a diverse one, indicating ongoing replication of the virus, and that ongoing replication was made possible because all of these sequences here had drug resistance mutations. So this virus was becoming resistant to the therapy that the patient was receiving at this point. This virus was not, and also every sequence of this virus that was seen was identical. And so this was one of these clones that we were talking about before. When the patient went on to therapy, this resistant virus, formerly resistant virus, went away. This therapy was switched to things that the patient was predicted to be sensitive to, and sure enough, it got rid of what we thought was a replicating population, showing that it was a replicating population, but this completely monomorphic, drug-sensitive population remained behind. And that was consistent with the idea that it was not a replicating virus. The virus was being produced by a clone of cells that had expanded greatly in this patient. So when we went back and looked at integration sites in this patient, we found that if you now put these on the same plot, again, this is the chromosome 16 map, that instead of having this nice pattern of integrations in genes, all the genes of this region, all the integrations were actually focused in one gene. And that's a gene called MKL2. And if we blow up this pattern here, so this is a very dramatic difference. We infer that in all the patients, this is the sort of thing we start with. This is what happens when you infect fresh blood cells, essentially. And this is what ended up, at least within this region in this patient. There were other proviruses in other regions, but this is the one that ended up here in this region. And if we blow this up, it's even more dramatic. If you look in the infected PBMCs at the integration sites in this MKL2 gene, you see they're scattered all over the place. And the different colors indicate different orientations of the provirus, either pointing this way or that way. And their random orientation, they're more or less random, although, in fact, as Dr. Levin has shown, there is a tendency of the proviruses to prefer the 5' end of the genome near the start site of transcription and taper off toward the 3' end. This gene, these little horizontal lines, vertical lines here are actually the exons, the coding parts of the gene. This gene is like most, is mostly intron with tiny exons. And when you look at the integration sites in this patient, instead of being distributed across, they're dramatically focused in this very small region. And if we blow up just that small region, which is only about 7 kilobases, the whole genome is about 3 billion base pairs. This is about 2 parts in a million of the total genome. And all of these integration sites, out of about 1,000 that we saw together, are there. The ones that are circled were actually seen more than once and are have to have been in cells that were clonally expanded. We think all of these cells were expanded. These are the ones that we could actually see as having more than one integration at the same site in different cells, for reasons I can't go into now for lack of time. We also saw exactly the same kind of thing in another gene, a gene called BOC2. BOC2 actually had previously been shown in a mouse experiment by infecting that mouse with another retrovirus, and that caused to be the cause of B-cell lymphomas, again with integrations in exactly the same region of the gene, mostly within this one rather large intron in this case. So, and again, some of these were amplified, and they were in this again in a very small region. It was a very small fraction of the gene as compared to what you saw in PBMC. So clearly there had been selection for, the only explanation for this is that there had been selection for integrations in this region. And this, if you're an old-time retrovirologist like me, this smells like what happens when retroviruses cause cancer, where you get integrations randomly, but then because some affect the expression of oncogenes nearby, proto-oncogenes, those then cause that cell to grow out as a cancer and a clone, basically. So HIV can not only be in clonally expanded cells, it can also participate in the clonal expansion of those cells, although it doesn't always do so. In many, I'll show you, there are many other examples where you get clonal expansion where you can't pin it on the virus doing anything because you only see it once. And those we think are where the cell itself has been caused to expand by antigen stimulation or some other means. So it has been thought that it had been thought up till this time. So I'll show you one more provirus in this patient. Clonally expanded HIV-infected cells will only harbor defective proviruses. That actually we have now falsified by the example I'm about to show you. So when we looked at integration sites, combining this patient and a couple of others, a paper we published a couple of years ago, we found that actually most of the integration sites, some of them were in this MKL2 gene, some of them were in a BOC2 gene, which is hiding down here somewhere. There were a lot of others at other sites, some of which were in unknown in sequences that were not known to be near a gene. But the one in particular is what we call AMBI because it was in a site that was somewhat ambiguous. That is to say that when we looked at the integration site sequence, it was present several times in the genome database, and we couldn't really tell which one. But we didn't really care which one because that's, I think, irrelevant to the point. So we cloned that sequence by PCR amplification because we knew the integration site. We could amplify the whole thing, starting with a primer from the cell DNA and going into the virus, starting with a primer from the cell DNA, going into the virus, and then we get 2 overlapping fragments, and this you could co-transfect into cells, and when we did that, we found, in fact, that those cells would make virus, and the virus was identical to the virus that we'd seen in plasma as being the prominent non-defective one, or non-resistant one, sorry. So this genome was non-defective, no deletions, no frame shifts, no stop codons, and indeed it was infectious if we transfected cells with it. And it was also the prominent virus in, this is now just a quick short summary of the patient sequence that I showed you, patient's history that I showed you before, just expanded. And this provirus, the sequence of this provirus was identical to the sequence of this prominent virus that we saw in appearing in blood. And we could even see a sample, the same sequence from a number of years before. So this was obviously in a clone that lasted a long time. It came up approximately, and we have a little more data on this that I'm not showing you, that it came up about as the tumor that was in this patient was growing. It decreased somewhat as the tumor was treated, and that led us to suspect. Okay, so first let me just say that if we take now PBMCs from the patient, activate those to make virus in an endpoint way, again, we find that at the endpoint, most of the virus that was produced by these cells, was done by John Mellors, a colleague at University of Pittsburgh, most of them produced a virus that was, again, identical to AMBI. So the majority infectious virus that could be induced with various chemical agents was also, again, this AMBI1 provirus. There were a couple of others that also came up that much rare, but that we haven't identified the viral DNA, the DNA for in these viruses. And that actually is a real problem with trying to study this, in that if you look at the DNA in the proviruses, the amount of the DNA sequences that correspond to the virus that you see in plasma are extraordinarily rare. Most of the time you can't find them. You can look at hundreds and hundreds of DNA sequences, and none of those correspond to what's in the plasma. The virus that you see is being made by a very, very small fraction of the total infected cells. Those are the ones, however, that you have to deal with if you want to think about curing infection. And I'm not going to tell you how we're going to do that because I don't know. So, and this is still true. This is the only known case where we can identify and characterize a clone of latently infected cells responsible for infectious virus in the blood. So it's in some kind of epigenetic state. We know a little bit about what might be driving clonal expansion. I said this patient unfortunately died. We were able to get some autopsy samples, although quite some time after after his death. Unfortunately, the pathologist doesn't get around to the Virgin Islands very often, and but what we found was that the virus was present, and in fact, cells containing this virus were present, and in fact were strongly enriched in metastatic lesions of this cancer, and you can see here just staining for CD4 on tumor-infiltrating lymphocytes, and you can see there are quite a few in this tumor sample. And with a good p-value, there was strong enrichment of these cells, and we hypothesized that the cells expanded because they were reactive to a tumor antigen. They were basically tumor-infiltrating lymphocytes that you may have, in fact, have heard about. So Mary Kearney, a colleague in Frederick, developed a very nice assay for looking not only at the DNA sequences in PBMCs, but also for pairing those DNA sequences with a level of RNA expression in each of those. I'm not going to go into the details on that. But what she saw is, so this is now a very small fraction of the total tree that was done by her and by Andrew Musick, a student up in Frederick. This is the AMBI1 clone, and what this shows is that these are the DNA sequences in this PBMC sample that she looked at. These are the RNA sequences that were being expressed by those cells. However, the DNA sequences was a very small sample compared to these. So in this sample, and we've actually expanded this to much larger numbers now, of 384 infected cells, only 4 of them had detectable RNA, and none of them at very high levels. The number of copies here is more or less corresponds to the number of copies of this RNA that were in these cells. So it's being expressed at very low levels in cells, only a few percent, and that we presume is how the cells can survive. That most of the time these cells This clone we estimate is expanded to about more than 10 to the 7th cells total over the whole patient, and has done so without, for the most part, the virus becoming expressed enough to cause the cell either to die by virus replication or to die because of the immune response. So our hypothesis from this is that the cells get infected, a small fraction. Now remember, we're talking about very small fractions of the total infected cells. There are 10 to the 12 cells infected every day, or 10 to the 9th cells infected every day, we estimate. Of those, a very small fraction enter some kind of a latent state where the cell can still grow, can still expand, be activated by antigen and so on. A very small fraction of these produce RNA spontaneously, and probably a much smaller fraction of these actually produce enough RNA at any one time to make virus. And so it's going to be a real challenge to be able to look at specifically these populations of cells. And these are the populations of cells you have to root out if you're going to try to actually consider how to cure HIV infection. It's a huge challenge. And so to summarize, integration patterns, I'm just the last part of this, integration patterns are very similar among different cell types. Integration sites can be readily identified in cells. This was a technical tour de force. It's only about 1 in 1,000 cells on average in these patients which have a provirus. And to be able to identify it in sufficient numbers to make conclusions about expansion and so on is quite remarkable. And that's thanks to a talented colleague, Shulin Wu, up at NCI Frederick. The integration site may itself influence persistence in clonal expansion, and therefore the virus that rebounds. Clonal variation in plasma can be mapped to individual integration sites. And expanded clones can contain infectious proviruses, and this was a matter that was under some debate until recently. And AMB1 clones may be expanded in response to tumor antigen. So I'll close here just to show you. I said that what I showed you was only a very small fraction of the total sequences. I feel obliged to show you the whole tree, but it has to be done in a certain way to really appreciate it. There's our friend Ambi going by. You don't need ultra-deep sequencing when you have good postbacks in the lab.
You show the correct tree from
Whoops. We can do it.
Well,
Okay, so I
thank you.
thank all these wonderful people, especially Mary Kearney, and Andrew Musick, who was responsible for the tree you just saw, along with lots of collaborators, both at NIH, South Africa, who did the baby studies, our colleague John Mellors at the University of Pittsburgh, and many others. Thank you.
Thank you, John. That was brilliant. May the force be with you. So we have time for some questions, and we urge you to ask anything that's on your mind. Don't feel that, particularly amongst students or fellows. Here's a student.
Learn something every day. So can you go into a little more detail about the the tumor cells you saw, did they have an integration event?
No, we don't think so. We've tried to look for that. I think, however, the possibility that HIV itself could be oncogenic remains to be investigated. If you talk to clinicians who studied HIV and studied HIV cancers, they'll poo-poo the idea, but I think it really needs some reinvestigation. The HIV tumor, T-cell lymphomas you'd think would be the prime candidates. T-cell lymphomas are not common in HIV infection. I don't think they're any more common than they are in uninfected patients. They're very rare anyway, so there's something about the biology of T cells compared to B cell that makes the lymphoma quite rare. But I also argue that the reason you don't see T cell lymphomas is if you get a T cell that's been transformed into some kind of precancerous state, that cell is now activated, is a very good target itself for HIV infection, and the HIV that's around at the time will just wipe it out. HIV doesn't infect B cells, you say. B cell lymphoma is the main disease for which, for at least some cases, we have no known viral cause for the increase that you see.
What about
But
neurological?
10 to the 9 cells are infected. T cells are infected every day with HIV. You tell me that 1 in a million B cells can't get infected. Yeah, I'll tell you, you're wrong, probably.
There's no association with brain cancers or neurological?
Not to my knowledge, no. There's, of course, other neurological issues.
So, John, are the long-lived cells the only reservoir for HIV?
I think so. However, there are still people who like to talk about macrophages, monocytes, dendritic cells, glial cells, other cells that can be infected, at least in the lab, with HIV. But
Well,
nobody has yet come up with any compelling evidence that these cells do actually form a reservoir. Nobody's got compelling evidence that they don't either, but there are some studies which really downplay the role of myeloid lineage cells in.
Well, do some of these long-lived cells actually reside in tissues?
Oh, sure. Yeah,
I mean,
most
are
of them
they
do.
all over?
Oh, yeah, they're circulating everywhere. Only about 2% of CD4 cells are in the blood at any one time. And so they're moving around fast. Contrary to what some people might say, there are people who like to say that, lymphoid tissue, for example, is a separate compartment from blood. We don't believe that at all, or gut. And we have studies that show that any given time, at least on patients on therapy, it's exactly the same virus population that's present in lymph node and in blood, or in gut and in blood. The cells move. There are some cases where there's some compartmentalization. If you look in the genital tract, for example, but that's probably because infection there is quite rare, and so they can be seeded sort of that side, and also the brain sometimes can be kind of seeded clonally. But over most of the body, the virus, in my opinion, is very well mixed.
So do you think there's any relationship to the distribution of the long-lived cells and the microbiome?
I don't know.
Well, I mean, there are studies
Sure.
showing that as you alter the microbiome, the lymphocyte population or
There are indeed such studies. Whether the specifics of what's in the microbiome and that sort of redistribution between the gut and the rest of the body actually has an effect on the HIV populations that are there, I think really remains to be seen. These might be studies that are better done in animal models where you can get better sampling.
What about an update about the public health standing? What number of infected people are there throughout the world, and how does that compare to 10 years?
Still, the number of infected people worldwide hovers around 30 to 35 million, of which, if I remember correctly, and somebody can correct me if I'm wrong, there are about 2 million deaths a year. There are about 3 million new infections, so probably still going up a bit. The death rate is going down somewhat, even worldwide, because of fairly good access and even in some rather poor countries to antiretroviral therapy, although there's still real issues with getting that used effectively, particularly monitoring the course of the infection, for example, the appearance of resistance, the ability to switch therapies, which you can do in the United States now. It's nobody who is diagnosed with HIV infection should die of AIDS. But it still happens. Unfortunately, it still happens at a pretty good clip, about 30,000 people a year, 30,000 new infections a year, and a few thousand deaths, I think. But everybody knows how not to get infected with HIV, and it shouldn't be necessary to spend billions of dollars to deal with it, but it is.
So, thank you. And there'll be time for more questions for Dr. Coffin or anybody afterwards. So, thank you, John. So, our second speaker is Jeffrey Lifson, who graduated from Northwestern Medical School, went to Stanford to take a residency in pathology. And while at Stanford, this correlated at about the time that HIV was emerging and associated with blood banks, where he was active as part of his pathology training, and that's how we got interested in HIV in the first place. He then spent several years in a biotech company developing new ways of looking at HIV, of understanding it, and came here to the NIH in 1995, where he is at Frederick, and his major interest has been in non-human primates as models of HIV and many other aspects of HIV virology, including some consideration of vaccine development. At present, he's a senior principal scientist and director of the AIDS and Cancer Virus Program of the NCI at Frederick. So thank you very much for being with us.
Yeah. Thank you, Win, and thanks for the introduction. As John told you, the more we learn, the better the science gets, the worse the prognosis gets for solving some of the remaining definitive problems for HIV. And those really are coming up with a way to cure infection. We have, for those that have availability to combination antiretroviral treatment, very good treatments. But as John indicated, those are not curative, and coming up with more definitive treatment that can eliminate the requirement for daily antiretroviral drug treatment is a current priority. Similarly, despite an awful lot of work by a lot of very clever people, we still don't have a vaccine that's capable of robustly preventing infection. And what I'm going to try and do today is to, not necessarily in the way of making excuses for my field, but just to give you as a non-specialist audience some perspectives on why it is that in spite of all the progress we've made, we still haven't achieved key milestones. So we'll demystify the title slide. And I'd like to start with a confession. You've heard the first part of the confession already, and that in the 12 years that this program has been operative, I have been always intrigued by it. It seems like a wonderful lecture series. I love the concept. I love the underlying premise. I love the idea of patient involvement. And somehow, I think partly because I work up in Frederick, this is the first time that I've made it here. So that's my confession. As I said, I think I love the concept of having a patient involved in these presentations. I have been involved in some clinical work in my past, and it's a really compelling part of it. And one of the things that makes my work so interesting is the translational aspect of it. With that said, as Win alluded to, these days, most of my patients are actually not people, but are actually non-human primates. And so I did consult with our veterinarians and asked them whether it might be possible to bring one of our SIV-infected rhesus macaques over to Building 50. They're just over in Building 14, and they decided that that really wasn't probably a very good idea. And so we'll have to accept a proxy for And then finally, I want to start with a disclaimer. As Wynn alluded to, I am actively involved in AIDS vaccine research, but it's not really the sole or even the predominant focus of the work that I do. But having said that, there are multiple people on this campus that for a specialized lecture on AIDS vaccine development, you have a whole building of them over in Building 40 at the Vaccine Research Center. And if you want to hear Barney Graham give a great talk on vaccine development, you can log on to the CROI meeting that was held a couple of weeks ago and check that out, the webcast. Larry Corey is going to be on campus in a couple of weeks. He heads up the HIV Vaccine Trials Network, and you can go hear Larry. But as I thought about this talk and initially felt a little bit uncomfortable about presenting on something that I don't necessarily consider myself an actual expert on, and then I realized that since this is a broad and very heterogeneous audience, that maybe it might be better to have someone that isn't going to get all down in the weeds on specific details of things. And so instead, the approach that we're going to take for this lecture is something akin to this, where I'm going to try and give you a perspective of someone who works in the general area. So it's very sophisticated cocktail party conversation sense of what the remaining challenges are in AIDS vaccine development and where the field is today. So vaccines depend on the concept of acquired immunity, which goes back to the 5th century BC, and found in a passage from Thucydides' history of the Peloponnesian War describing the plague of Athens. And the pathogen responsible for this plague is still not entirely clear to scholars, but what was clear and very vividly accounted by Thucydides is in this passage. Yet it was with those who had recovered from the disease that the sick and the dying found most compassion. They knew what it was from experience and now had no fear for themselves, for the same man was never attacked twice. So this is the first historical example depiction of what we understand as acquired immunity. Obviously, the underlying scientific concepts in terms of the basis for acquired immunity was not well understood at the time, but this is really the first understanding of it, and vaccines are based on this premise. Having subjected you to Thucydides and the history of the Peloponnesian War, I'm going to skip the otherwise obligatory Jenner cowpox slide and move on to say that over the succeeding centuries, we have come up with all kinds of vaccines, and this credit roll is just a partial listing of the vaccines approved by the FDA for use in the United States. And there's a long, long list of very successful vaccines. Having said that, I'm going to make the case that in contrast to the situation for HIV, all of those successful vaccines, and this is not in any way to downplay or minimize the difficulty in coming up with those vaccines or the contributions of the people that have come up with clever ways to generate those vaccines. But to some extent, all of those successful vaccines represent a combination of a triumph of successful empirical testing, a combination of luck, and fortuitously tractable targets. And as I'll probably convince you by the end of this presentation, none of those things seems to apply for HIV. As an example of that, this is a photograph from a very famous press conference from almost 33 years ago. That's a young Bob Gallo and then Secretary of Health and Human Services, Margaret Heckler, who famously gave a long extended quote. First, the probable cause of AIDS has been found, a variant of a known human cancer virus called HTLV-3. That's changed. Second, not only has the agent been identified, but a new process has been developed to mass produce the virus. Discovery is crucial because it enables us for the first time to characterize the agent in detail and understand its behavior. Discovery of both the virus and the new process. We have a blood test. We've applied for a patent. With the blood test, we can identify people. We can ensure blood is for transfusion is free. We should be able to prevent transfusion-related cases, diagnose people, potentially treat them. And we also believe that this will allow us to develop a vaccine, and we hope to have a vaccine ready for testing in approximately 2 years. That was in the spring of 1984. Now, Margaret Heckler gets a lot of crap for that quote, which sometimes gets taken out of context because I think people read that quote and interpret that quote as we're going to have a vaccine that prevents HIV infection in 2 years. And that wasn't what she promised. She said that we would have a vaccine ready for testing. And we didn't miss it by that much. It's just that the vaccine that was ready for testing wasn't very good. And the vaccines that were initially tested, it's hard to go back to this. And, we're going back 30 plus years. I've been doing this for longer than looking around the room. Some of you have been around. But that said, the initial vaccine development efforts was taking place in an environment where I think the vaccinology field was flushed with the success of initially purified and then ultimately recombinant subunit vaccines that were successfully preventing hepatitis B infection. And people thought, gee, this is terrific. We have this whole new technology. All we need to do is sort of in a turnkey fashion, let's apply the same thing that works so well for hepatitis B to HIV, and that'll solve the problem. And long story short, it didn't come anywhere near close to solving the problem, which turned out to be much more complicated. Somewhat surprisingly, over the intervening 33 years, there have really only been a total of 6-ish studies that have been conducted at a large enough scale, either phase 2B or later, with the potential to identify potential efficacy of a vaccine. The first two were just a recombinant subunit approach that were sort of dismal failures. The next two were related trials trying to do a cellular immunity response using a recombinant adenovirus approach, focusing on trying to induce protective cellular immune responses. We'll talk about that in a little bit more detail in a minute. And then the one sort of partial success is the RV144 study, so-called TIE trial, that showed a marginal estimated about 31% efficacy, and intriguingly was a study that was based on a study that a lot of people thought should never have been done, based on combining two components, neither of which worked very well together into something that worked at a just barely detectable level. And now there's currently a large study going on in South Africa trying to evaluate something that's probably best described as an extension of the underlying approach that was used in that TIE trial. But there haven't been a lot of efficacy trials, in part because the results from earlier stage clinical trials and preclinical results haven't been encouraging enough to justify large-scale clinical trials. So why is that? Let's talk about some of the underlying conceptual premises of what has enabled successful vaccine development for other targets. One thing that's important to realize is that a lot of people think about vaccines and there's sort of a simplistic notion that, well, you vaccinate someone and that prevents infection. Most of the time that actually isn't true. What vaccines typically do is that they control and clear infection and prevent it from becoming clinically manifest. And that's an important distinction because for some pathogens, that's a perfectly acceptable outcome. But if you were paying attention to John's presentation, you realize that HIV is a sneakier pathogen than many, and one of the things that it does is to integrate into the chromosomes of some of the cells that it infects, and it can establish a long-lived state there that doesn't go away until that cell goes away. So, simply controlling the infection, which we can do quite effectively with antiretroviral drugs, a vaccine response that does only that may not be enough. Similarly, for most of the things for which we have effective vaccines, there are clinical examples of basically naturally occurring or post-convalescent immunity, the type of thing that Thucydides was talking about. Individuals that survive become refractory to reinfection. And in many cases, we can understand an immune correlate of that state, either at the level of something that is a causative correlate, that is to say, by an interventional study, you can show that a particular response is responsible for that protection, or a surrogate marker of protection, whereby you can show that it may be that measuring this particular antibody response may not be directly involved in protecting against the pathogen on rechallenge, but it correlates well enough that you can use that as a reliable marker for whether or not your vaccine is likely to work. And finally, animal models have been critical to the development of a lot of vaccines for HIV. I've spent the last 20 years trying to develop better animal models. HIV itself doesn't readily infect other species. And so what we do is work with simian related viruses that recapitulate many of the key aspects of HIV infection in humans. We can generate chimeric viruses that are part HIV and part the simian immunodeficiency virus, or SIV, that infects monkeys. And more recently, with some of my colleagues up in New York, Paul Binoche and Theodora Hatziana, we've been generating minimally chimeric HIVs that have just minimal changes. They're 95% HIV sequences, but they're able to productively infect macaques and cause disease in those animals. So there's room for expansion and improvement of the animal models we use, but they're better than available for some diseases, for example, hepatitis C, but not as good as some of the models that are used for other targets. It's not that we don't have a vaccine for lack of trying. This is just summarizing a lot of the various different vaccine approaches that have been evaluated, either classical whole inactivated, or live attenuated, or subunit, or synthetic peptide approach, including synthetic peptides packaged in various different creative ways with nanoparticles and everything else, recombinant viral vectors, recombinant bacterial vectors. DNA immunization has been evaluated, and most recently people are starting to use, some of you may have seen a recent publication involving the use of RNA for Zika virus immunization. Those approaches are also being evaluated for HIV type vaccines. All of these things have been tried with a lot of creativity without a lot of notable success. Why is that? So there are a number of factors, and in combination, they represent a daunting set of challenges for vaccine development. We've touched on the fact that one of the paradigms for coming up with a successful vaccine is having examples in nature of where an individual has been able to survive the infection and establish a state of refractory immunity. That's not the case for HIV. We don't have examples of people that become infected, are able to survive and spontaneously clear the virus. And in fact, we have only limited examples of people who, by virtue predominantly of having some particular MHC alleles that present viral antigens that are particularly required for viral replication, are able to control the virus very effectively, so-called elite controllers that in the absence of a therapy are able to keep their viral replication levels down quite low. But even those individuals are not the equivalent of being uninfected. John mentioned the replication properties of HIV. So HIV is a retrovirus. That means it replicates by turning its RNA genome into DNA and then integrating that DNA into the host chromosome. It does that RNA to DNA step with a reverse transcriptase, which is an enzyme polymerase that does not have a proofreading function. And that means that it's a relatively, compared to the cellular polymerases, an error-prone process. And so you can think of HIV as having adapted a life strategy of saying, whatever environment I find myself in, I'm going to generate lots and lots of variants. And in whatever environment I find myself, I'm going to come up with some variants that are going to have a selective advantage in that environment, and those will proliferate and expand and take a, and if the environment shifts, it'll change again. That generates tremendous diversity and the capacity to escape immune responses, either in the course of natural infection, both antibody and T-cell responses, or conventionally generated vaccine responses. HIV attacks and eliminates CD4 cells, which are critical for coordinating immune responses, and also in various ways disables other key immune cells. The virus gets out ahead of the host and never gives up that advantage. The immune system winds up chasing after viral replication. The structure of the key antigen for antibody-mediated neutralization for HIV is conformationally very complex and is extensively glycosylated. More than half of the total glycoprotein is glycosylation, which makes it difficult to generate antibodies to the targets that you'd like. And then, as John dwelt on quite a bit in his presentation, once the virus gets in, it integrates and can establish latency and various other long-lived persistent forms. All those are challenges. This is mostly for John's nostalgic value. This is an old slide, but it illustrates the point nicely. We tend to think of influenza as a virus that varies quite a lot, requiring a different vaccine every year. This shows for 1996, the global, this is a phylogenetic tree showing the extent of diversity in the global influenza in 1 year. Through that diversification process that John talked about, generating variations in a single patient over the course of a few years of infection, you can see that the extent in an individual is comparable to the global influenza variability. If you look at a relatively small cohort of individuals in one city, it's much bigger. And if you look in one country, you can get a sense of the relative scale. So there's tremendous diversity, which is a big challenge for coming up with a vaccine when you're trying to come up with something that can be universally applied. So when we think about mechanisms of vaccine protection, we don't really know what it is that we're trying to go after in terms of what is a protective response, because we don't have that example of somebody that has come through natural infection in a state that resists reinfection, that we can say, what is the nature of the responses that allow that? So we can look at, well, what are the potential things to look at? And on the antibody side, we can look at neutralizing antibody. But because I showed you all that diversity, we need to worry about not only the potency of antibodies, but also the breadth, their coverage of all these diverse isolates. We can also talk about maybe non-neutralizing antibodies that may have activity through Fc receptor-mediated functional activities like targeting other effector cells for antibody-dependent cell-mediated cytotoxicity or other effector functions. We can talk about T-cell immunity. We can also talk about trying to enlist the innate immune system through various different adjuvants. When we think about, what are some of the approaches that we can use vaccine-wise in terms of the nature of immunogens, just to reiterate, most of the regimens that people are using are some form of prime boost, where we stimulate in a couple of different ways and multiple immunizations to stimulate those responses, either using a killed antigen subunit. Currently, the best immunogens for envelope glycoproteins seem to be so-called SOSIP trimers that start finally, after a couple of decades of intensive work, now are starting to have immunogens that more authentically recapitulate the structure of the native antigen. And various live vectors, both replication incompetent and competent, nucleic acids. But one of the things that people have come up with approaches to deal with that diversity that we've talked about is if you just take a single prototype sequence as your vaccine insert, that's not going to likely cover the entire range of diversity that's out there in terms of all those different viruses. So what people are focusing on are using either consensus sequences based on large databases of sequenced viruses that have been accumulated, or conserved regions of the viral genome that are presumably conserved because of functional constraints that are obligatory, or perhaps most creatively and most recently, coming up with so-called mosaic vaccine inserts, which are basically in silico predicted epitopes, which may not even exist in the real world, but basically are algorithmically generated based on all of the diverse sequences to provide induction of responses for maximum coverage. And those mosaic inserts actually do turn out to give a few-fold increased reactivity across a broad range of diverse isolates. Also importantly is the immunization scheme that's used. Conventional immunization schemes are unlikely to give successful immune responses for reasons I'll get into in just a minute. I'll say I've been doing this for a long time, and the field has swung back and forth between approaches that emphasize trying to induce humoral immunity and antibody responses and cellular immunity. I think as with most questions having to do with HIV, the answer to most either-or HIV questions is yes, probably both, and that's probably going to be true for a successful vaccine approach here. But let's start by considering neutralizing antibody-directed vaccines. This is an area that I follow. It's not an area I work in directly, and so I'm indebted to some colleagues, Bart Haynes and especially Dennis Burton, not only for sharing some pre-publication information with me, but also some of the slides I'm going to show you. The neutralizing antibody approach is premised on the fact that the way that HIV infects cells is by the envelope glycoprotein, shown on the bottom here in the purple and green, interacting with a receptor, the CD4 molecule that's involved in immune regulation responses, and a chemokine receptor, typically CCR5, although a minority of viruses can also use a different alternative chemokine receptor called CXCR4. So these two molecules need to interact. This envelope glycoprotein is a trimer of heterodimers. And we'll get into the structure of that a little bit more. But what happens is, as with most enveloped viruses that have an envelope glycoprotein, it engages a receptor that triggers a conformational change in what is essentially a spring-loaded, conformationally flexible molecule that results in exposure of a hydrophobic domain that then mediates fusion of the viral envelope membrane with the cellular membrane, allowing viral entry of the capsid core and the subsequent steps of the replication leading to integration and infection. So there's this multi-step process of engaging the CD4 receptor, the co-receptor, and getting the virus into a cell. Basic premise is that neutralizing antibody, shown in red, can bind to the envelope glycoprotein and basically sterically block that interaction and keep it from happening. We're in building 50, so I'm going to show an obligatory slide from my colleague Sriram Subramaniam upstairs. And this is using a technique called focused ion beam scanning electron microscopy, or also known as ion abrasion electron microscopy. And basically what you can do is take a relatively large, for electron microscopy, specimen, fix it, and with a dual beam microscope, in which one beam is a conventional imaging electron beam, but the other beam is an ion beam that basically vaporizes a thin layer off the top of your sample. You can take a picture, vaporize a layer of 10 to 25 nanometers, take another picture, and so on, then integrate all of those images into a three-dimensional composite image that allows you to get a three-dimensional perspective on mixtures of cells or tissues. So it's a powerful technique, and what that shows you is that rather than simply having to block the access of free virus to a cell floating in a nice culture flask, in vivo the situation is likely to be much more complicated when cells of the immune system, CD4 cells, particularly interacting with dendritic cells and other antigen-presenting cells, have these extensive membrane interactions and interdigitations. And in fact, if you look at an interaction between a dendritic cell and T cell, you can see, focused in red, the virus which is being transferred at what's known as a virologic synapse. And again, you can see that even things that look like just thin cell processes here in the 3D reconstruction are actually sheets of membrane. And if you think about trying to get access for an antibody to disrupt that, that's even harder than if you're trying to block access of a free virion to a target cell. And so there are many challenges where as John alluded to, the evolutionary adaptation of this virus has basically subverted the natural physiology of the immune system and interactions of cells in the immune system to its own advantage. There's another challenge. I've told you how hard it is to generate neutralizing antibodies. Well, there are some, as we'll get into in just a minute, and they work well. One of the ways that we've been able to validate that they can work well is in non-human primate models. But rather than the simplistic notion that I and a lot of people had before we started doing these experiments, that a neutralizing monoclonal antibody is going to act predominantly by neutralizing the virus at the portal of entry and preventing infection, it turns out that that isn't the case. And in a recent study with Dan Baruch, Dennis Burton, and Rafiq Sekeli, what we showed in animals that were treated systemically with the neutralizing antibody PGT121, don't worry that you can't read this. This is basically across the way, lots of different tissues that were surveyed in a serial sacrifice study in monkeys on day 1 through 3, day 7 or day 10. And the tissues highlighted with the blue backgrounds are tissues outside of the, in this case, following vaginal inoculation, the female genital tract port of entry. These are all distal tissues in blue. The pink represents the genital tract. And you can see, compared to sham-treated animals, the animals that got the antibody, highlighted here, actually had more virus at distal tissues at early time points, but they eventually went on to clear that virus. So it's not a simple case of blocking things at the portal of entry. It's much more complex than that. And it wasn't just that we were picking up, stray non-replication competent nucleic acid signals in these distal tissues because we're actually able to transfer infection by adoptive transfer of cells from these distal tissues at early time points. So the biology of this transmission is much more complex than we've appreciated. So neutralizing antibodies need to target the envelope spike. As I said, that's a trimer of heterodimers, and we have developed a really extensive knowledge of its structure and functional properties, and that has helped to guide, but hasn't led us to completely successfully solve how to make a vaccine with the desired properties, as I'll get into. This simply shows a crystal structure and a cartoon structure where the 3 transmembrane envelope glycoprotein subunits are non-covalently associated with the surface envelope glycoprotein subunits. You can see that in this model here with the subunits showing as we rotate around. And if we turn to a space-filling model where the green is the GP120 with the yellow highlighting the CD4 interaction domain, and the red is the transmembrane envelope glycoprotein, you can appreciate the structure of that. And when you add in the glycosylation, you can see how obstructed that structure really is. So there's a tremendous challenge to generate antibodies to the kinds of vulnerabilities that we would like to be targeting with a vaccine. That said, we've made a lot of progress. A lot of that progress has been enabled over the last decade or so by studying naturally infected people that have developed antibodies over time. And what typically happens when someone gets infected is they'll initially develop some antibodies that bind to the envelope glycoprotein, but those antibodies typically don't neutralize even the virus that they got infected with. By about a year or so, they'll start to develop so-called strain-specific antibodies. That is, antibodies that can neutralize the initial virus that they were infected with and that was present through that first year. But by the time they've developed those antibodies, the virus has moved on and mutated so that that antibody no longer neutralizes the virus that's actively replicating. That process goes on in a way that kind of reminds me of the coyote chasing Roadrunner and never quite catching up because the virus keeps mutating to stay at least one step ahead of the antibody response that's chasing it. But after multiple years, a subset of patients, and depending on the cohort, between 5 and 20% do develop antibodies that are capable of broadly neutralizing a range of different HIV isolates. Unfortunately, it doesn't do them much good because, again, the virus is multiple steps ahead and has mutated, but we've been able to learn a tremendous amount about what are the characteristics of antibodies like that. And that's really been enabled by a technique that's been pioneered by Michelle Nussenswag at Rockefeller and Dennis Burton at Scripps, in which they've kind of taken sort of a reverse vaccinology approach where they start with the antibody and then do structural studies of that antibody interacting with various model envelope antigens, and then try and glean from that information to help guide the design of new immunogens and then test those immunogens for immunogenicity. And this has really been facilitated by techniques that were worked out to use fluorescently labeled so-called baits, where you use your GP120 antigen fluorescently labeled to stain individual B cells, then are able to sort out the individual B cells and clone the B cell receptors from those and generate, through molecular techniques, neutralizing antibodies. And there's been tremendous progress over the last several years in this area, and lots of antibodies have been developed. Some prototype antibodies are shown here, where this is the GP120, GP41 trimer of heterodimers with the virion envelope membrane here. And there's one class that are called apex antibodies, typified by PG9, some that interact with what's called the high mannose patch. We showed all that glycosylation a while ago. That acts as an impediment to the binding of most antibodies, but there's actually a subclass of antibodies that are dependent on the mannose patch for their reactivity. CD4 binding site antibodies, things that bind at the interface between those subunits, and things that bind in the so-called membrane proximal external region, which, as you can see, is a tough, tight fit, but there are antibodies that are able to get in there. This just shows a table showing the relative potency of the antibodies for in vitro neutralization. relative to the percentage of a large panel of roughly 100 different diverse virus isolates that they're able to neutralize. And this kind of composite gives you an idea. These were some of the initial antibodies that were identified to those different domains. And we're actually making tremendous progress here. And now we have a number of antibodies that are actually reasonably broad and quite potent. As we've studied those antibodies, though, and as you might guess, based on the process that I described to you of how they arise and how long it takes, what's become clear is that most of these antibodies are not your typical garden variety antibodies. They're really unusual. They have unusual characteristics that are just not typical for most antibodies, including extended lengths of the complementarity determining regions for some of them. That is, they need extra long binding domains to access their target antigen. And part of that is associated with a requirement for extensive somatic hypermutation that comes about in the course of maturation of the antibody responses. So that's part of the explanation for why you need such a long time to develop these antibodies, because you need to accumulate lots and lots of mutational changes in order to come up with the properties that are required. So we have a tremendous problem. We have these wonderful structures of antigen bound to antibodies isolated through the process I just described to you, but we're still kind of stymied in how to translate that information into how do we come up with an immunogen that we can immunize people with and then generate those antibodies de novo in a practical way. So that's bridging the gap from antigen to immunogen. And so the things that people are working on now is trying to generate more conformationally authentic trimers, and we've made a lot of progress over the last few years with that. One of the challenges is because of you need that maturation. It turns out for many of the lineages of antibodies that have these desirable properties, the final antibody that you want, the final structure that you're trying to engage, the B cell repertoire doesn't contain something that binds to that. You need to bind to something different than that and then go through the sequential mutation process to get to where you need to. And so to deal with that challenge, people are starting to do some sequential immunization approaches where they immunize first with one simplified version and the next version and so on, 3 or 4 different things. And whether we'll get there with that approach or not, I don't know, but it certainly says that just a conventional immunization, the way that we're used to for other kinds of pathogens, isn't going to work here. And then finally, as you might figure from how long it takes to develop the kinds of antibodies in the course of natural infection, where you have continuous exposure to large amounts of antigen to drive that process, simply giving a couple of shots at 6-month intervals isn't going to get us there. So understanding the immunization schemes that are going to be required is going to be important. So as we continue to work on that challenge, one of the things that the field has adopted is to say, well, we don't know how to generate these broadly neutralizing antibodies from immunogens just yet, but we do have all these broadly neutralizing antibodies that are potent and broad. Can we both learn something in terms of proof of principle about the potential of broadly neutralizing antibodies once we figure out how to induce them through a vaccine, but also have some practical impact by using passive administration of such antibodies? And there are clinical studies in progress now to test that proof of concept and see whether we can impact transmission in both pediatric and adult high-risk cohorts. It's likely that, as with most things, monotherapy with drugs or antibodies or anything else is often a recipe for generating resistance. So we'll likely need multiple antibodies, even for a prophylactic application where the challenge inoculum is typically relatively small. And of course, recombinant proteins like antibodies can be quite expensive, but there's been a lot of progress recently in engineering antibodies with certain changes that dramatically extend their circulating half-life from a few weeks to months. And so there's progress there. And one other approach is the use of gene therapy approaches, in particular adeno-associated virus encoding these neutralizing antibodies. And Mike Farzan and Ron DeRozier in non-human primate studies that we've collaborated with have very encouraging results on the potential of AAV-delivered antibodies to either prevent and or treat infection in non-human primate models. I'll touch briefly on non-neutralizing antibody targeted vaccines. This is I alluded to the RV144 TIE trial. The conceptual paradigm for the potential importance of these non-neutralizing antibodies comes from that study, which, as I said, had really borderline efficacy at 31%, but that efficacy was not associated with either conventional CD8 T cell responses or neutralizing antibody responses. Instead, it was associated with CD4 responses to envelope glycoprotein and non-neutralizing ADCC-mediating antibodies, but only in the absence of IgA responses to the vaccine, which actually seem to have a negative influence on protection. I'm not a big believer in the utility of these kinds of antibodies. I think really the only vaccine I'm aware of where there's a clear case that non-neutralizing activity plays a key role is for pneumococcal vaccines. But it's something that is being actively pursued in the field, and now you know about it. Let's shift now to looking at cellular immunity targeted vaccines. As I said, the pendulum has swung back and forth, and in particular, one cellular-based vaccine after the initial failure of the first subunit antibody targeting vaccines was an adenovirus serotype 5 vectored vaccine that really sort of killed the whole field of cellular immune targeted vaccines for a while because, as you can see here, in study participants that were Ad5 seropositive and had pre-existing immune responses, the vaccine actually seemed to produce an activating response that enhanced susceptibility to infection. And so obviously that was discontinued, and enthusiasm for cellular-based vaccines in general suffered as a result. I want to spend my last few minutes here talking about a completely different kind of vaccine, and this is work from my longtime collaborator Louis Picker out at the Oregon Health Sciences University and the Oregon National Primate Research Center. Louis is one of the people that developed the technique of flow cytometry technique of intracellular cytokine staining, and he used that several years ago in a technical tour de force of basically using thousands of peptides to map the complete immunological repertoire of responses against cytomegalovirus. And that's just represented here as a classic paper. But one of the interesting things about those responses is that they're very broad, but they also have some other characteristics. In particular, they're skewed towards effector memory differentiation state rather than memory. So when you do a conventional vaccine, and this is just simply showing a hypothetical curve of the time course of viral load in plasma as a consequence of viral replication, in a naive individual, by the time the cellular immune response is peaking, you're multiple weeks into the infection. Even in a conventionally vaccinated individual, because in a prime boost vaccination, typically what happens is you get a peak response and then that dies down to a memory state. Then you need anamnestic proliferation and expansion of that response, trafficking of those cells to the appropriate sites before you get a peak response. Even there, you have a significant delay. And it looks like for HIV, that delay is just too damn long. There's a shorter window of opportunity. And so we ask the question, if CMV, which induces these effector memory responses that are already differentiated and ready to go and persistent because the vector CMV is also persistent, could that have a beneficial effect? And so we tested the idea of whether or not these CMV vectored responses, they give very broad CD4 and CD8 responses, lots of diversity of the epitopes that are recognized. Because it's a persistent vector, it gives persistent responses that are skewed towards effector memory and have a broad tissue distribution, including to sites of mucosal challenge. The question was whether using CMV as a vector would give responses to the SIV inserts in these non-human primate studies that had these properties, and if so, would that be associated with vaccine protection? And the short answer from many, many studies is yes, of a kind, and really of a unique kind. And what you're seeing here is unvaccinated animals challenged intrarectally for the data shown here. And you can see the plasma viremia curves for the animals. And what we saw is that in this study, this vaccine does not induce appreciable antibody responses to the inserts. It just induces these really interesting T cell responses. And those responses aren't able to prevent infection. But what is striking is in over half of the animals, they're able to control infection and durably control infection over time. And what's even more striking is that when we look longitudinally in animals, you can see that we initially see these plasma blips that decrease and go away. Initially, and I'm showing you RNA and DNA levels in bone marrow, but in multiple other tissues also. Initially, in animals, as soon as they clear plasma viremia, we can show that there's still replication competent culturable virus in these tissues. There's RNA, there's DNA we can adaptively transfer, but over time, we can no longer detect these things. So these animals are progressively clearing the virus from an early established multiple tissue sites. And what we think is going on is, as John was alluding to in his talk, there seems to be a transition from those short-lived cells that are infected to long-lived cells, and we don't fully understand anatomically or cell compartments or whatever what that transition is. But if the infection can be contained before you cross that threshold, it appears to decay over time and to eventually be cleared. And that's shown here. If we look at animals and looking at DNA levels or RNA levels in these various different tissues, in animals 3 to 6 weeks after challenge, they've become aviremic and then were sacrificed and necropsied and tissue studied. We can still see readily detectable RNA and DNA. In contrast, if we follow animals out for 2-3 years, we can no longer find anything that is detectable by laboratory analysis or adoptive transfer. So we're seeing progressive clearance of virus from these tissues over time. And just to make it a little bit more interesting, I started out my training wanting to be a cellular immunologist. I got diverted for a couple of decades doing AIDS-related stuff. Now I'm getting back to studying cellular immunology, but it's a variation on cellular immunology because it turns out that this vaccine induces a very strange kind of cellular immune response. Ralph Zinkenagel and Peter Doherty got the Nobel Prize in 1996 for their work in the 70s demonstrating in murine systems that T cell responses by CD4 cells were restricted by MHC class II antigen presentation, and responses by CD8 cells were restricted by MHC class I antigen presentation. And that's still true, but it's not the whole truth, because it turns out that the responses induced by these vectors, actually the CD8 responses, rather than being conventionally restricted by MHC class I, about 2/3 of them are restricted by MHC class II, and the rest are actually restricted by an allele called MHC class Ie, which is much better known for being involved in regulating NK cell responses. It's a long story, and some of it's published, some of it is in preparation. It looks like it is a consequence of the fact that this vector that we used was passed in fibroblasts for a period of time and acquired some deletions that resulted in altered host cell tropism. And as a result, in vivo, it has altered host cell tropism, and the antigen priming and presentation is quite different than other vaccines, natural infection, or even wild type CMV infection, all of which would be interesting, but a side note, except for the fact that when we make repaired vectors and put the mutations back and test the various combinations, it's become clear that these unconventional responses are actually what's responsible for the protection that we're seeing. So we're trying to get to the bottom of that. So that brings us to the final chapter of our HIV vaccine development for sophisticated, highly intelligent, but non-specialists. And I'll leave you with a couple of take-home messages. Hopefully I've given you a sense that coming up with an effective vaccine for this very wily and sophisticated target pathogen represents extraordinary and to some extent unprecedented challenges based on both its properties as a target, but also the biology of the pathogen. I hope I've given you a sense that we haven't been wasting our time. We've made a lot of progress, but an efficacious vaccine really remains elusive. My conviction is that we've tried all the easy and obvious stuff over the last 3 decades, and that a vaccine solution to this will yield, but it probably is not going to be based on any kind of a conventional immunogen and/or vaccine approach. But with all of that frustrating stuff said, I think the pressing need for a vaccine and the potential global impact of even a partially effective vaccine is so important that it continues to motivate me and others in the field, and I think to justify continued research in this area. I will stop there, and if we've got time, try and answer any questions.
Okay, thank you very, very much. That was marvelous. So I trust you have some questions you wish to ask? Yes. Here, use the microphone. So.
Thank you so much for the really nice talk. Could you elaborate a little bit more on the possibility of us really getting a prophylactic vaccine versus a therapeutical? And in terms of timeline, what do you see?
Yeah. So John alluded to the situation of persistent virus in the face of antiretroviral therapy, and people are pursuing a number of different approaches to try and come up with more definitive treatment in that setting. Some of those approaches involve so-called therapeutic vaccination, where the idea is to augment immune responses, and oftentimes that is premised on a combination of an immune clearance mechanism that is joined to some kind of a stimulus that's intended to induce expression of latent virus, because without expression of virus, that persisting virus is basically silent to the immune system. It's not detected. I think that's a really daunting challenge, in part because the amount of virus that's there is higher than what you need to prevent in the initial prophylactic setting. Whereas John alluded to, the molecular data suggests that most transmissions involve transmission of a single viral variant across a mucosal surface. So that, in principle, is less of a challenge, and so on. I hazard to give you a guess about timelines for vaccines able to be effective in either setting. I think there are huge challenges. I think some of the work with passive antibody that's going on will demonstrate proof of concept, hopefully, for particularly neutralizing antibody. And there may be some practical applications for that before we have immunization-based approaches for prophylaxis. In terms of therapeutic vaccines, it's become clear both from clinical work and some of the studies that we and others have done in non-human primate models, that to be effective, you either need to have completely effective immune surveillance that is going to control whatever virus is left when you take the antiretroviral drugs away, or you need to actually eradicate every last replication competent provirus. And that's an extraordinarily challenging bar to get over.
Henry?
But like John said, the more sophisticated our knowledge becomes, the more daunting the task seems to get. But it's a progress of a kind.
Jeff, that was a beautiful talk. Thanks for all the background. Can you say a little bit more about what could be the explanations for a short-lived infected cell versus a latent cell? Does it not have anything to do with selection of the integration site or expression of the virus into chromatin?
I think that there are probably multiple things that can contribute to that. I think integration site may contribute to persistence. The one thing for which there's actually some data that are relatively clear is that if you look at the distribution of proviral DNA in different CD4 memory differentiation subsets, in people that go on treatment or animals that go on treatment very early in the course of infection, most of that DNA is in shorter lived cells. It seems to not get into central memory cells that are longer lived and can establish latency just proportionately to the same extent. And that's one possibility. Another possibility that we're looking at has to do with the fact that there are a number of different immune privileged tissue sanctuaries for virus. One of the ones that we focused on is actually T follicular helper cells inside B cell follicles, which for various reasons, cytolytic T cells that are able to control and clear virus from other compartments because of the natural biology by which they're not able to effectively traffic into the follicles. That remains a viral reservoir. It could be something where whether or not you've crossed a threshold in terms of virus getting into those kinds of sanctuary sites or not may make a difference in terms of establishment or not establishment of a long-lived viral reservoir.
And what typically kills a cell that's infected by HIV? Is it just over replication, or is it?
So it's clear that in vitro replication and viral cytopathology can kill cells, including primary cells. The catch there is that in vivo, to the extent that we can quantify viral burst size and so on, it seems to be much less than under maximally stimulated conditions that we use in vitro in culture. And so with the so-called latency reversing agents that have been used so far to stimulate cells, both in vitro and in vivo, the initial hope of the so-called shock and kill paradigm was that you could stimulate cells, you'd get induced expression, and either you get so much virus expression that the cell would die from viral cytopathology, but even if it didn't, there'd be enough antigen expression that you'd have immunological clearance. Shock and kill has so far kind of turned out to be tickle and hope because the level of viral induction that you get doesn't seem to be enough to actually result in viral cytopathology. And under normal conditions, the majority of the cytotoxic T cells that are around aren't able to kill those cells, even if you do get a little tickle of viral expression.
So I would mention to you that all of the PowerPoints and reference materials are all available on our website. All you have to do is Google demystifying medicine. In fact, you can see materials for all of the programs for the past 12 years. You can follow the evolution of HIV by looking at that website too.
You can binge watch Demystifying Medicine.
So listen, thank you very much. This has really been extraordinarily exciting. Thank you.